Hello, everyone. My name is Magda Markowska. I'm a PhD student at University of Warsaw, and I would like to present CONET, Copy Number Event Tree Model of Evolutionary Tumor History for single-cell data. First, I would like to explain why we need to model copy number alterations. Copy number alterations are ubiquitous across all types of cancer. They are also the most common alteration associated with tumor hypermutability. Moreover, they can be important drivers of tumor evolution. Deletions can deactivate tumor suppressors, and amplifications can activate oncogenes. Recent developments in single-cell sequencing have achieved relatively even coverage across the whole genome, allowing us to call copy number profiles for single cells. But so far, there have been no methods published for inferring copy number evolution from this type of data. Here on the left, you can see single-cell DNA sequencing data in the form of corrected counts per beans. These counts were corrected for GC content and mapability, and they were also normalized so that 2 means neutral copy number. We call this type of data per bean data. You can observe that the corrected counts per beans follow mixed normal distribution, with the most prominent component of this mixture having expected value equal to 2. The second most prominent component has expected value equal to 1, and we can draw a conclusion that probably in this sample we deal with quite a lot of deletions. We can further transfer this per bean data into per breakpoint data when we calculate the corrected count absolute differences in adjacent beans. So this data is given not for genomic beans, but for genomic loci. We can divide these absolute differences into 2 subgroups. The red subgroup is calculated for loci where we do not expect copy number change. So this is no breakpoint loci distribution. And the blue subgroup is calculated for loci where we expect copy number change. So we call it breakpoint loci distribution. As we can expect, the red distribution has expected value equal to 0 and quite a low variance. And the blue distribution is again a mixture of different distributions, with definitely the most prominent one, corrected count absolute difference equal to 1. This means that the most popular copy number change is probably by 1. What is also worth observing here is that those 2 distributions overlap, and this overlapping represents the noise in our data. In panel A, you can see graphical illustration of data preprocessing step. Colored circles mean single cells, and gray shapes mean genomic loci. First, we have the corrected counts data matrix. This is our per bean data, and genomic loci have a start locus and an end locus. This data is further transferred into corrected counts absolute difference matrix. So this is our per breakpoint data, when in columns we have genomic loci instead of genomic beans. The third part of our input is potential breakpoint set. Before we start our modeling procedure, we need to decide which genomic loci do we want to include as possible borders of copy number events. Okay, so let's start our modeling. First, we need to construct a copy number event tree. This tree always has healthy cell as a root, and then each node means 1 copy number event. And the copy number events are denoted as a start breakpoint and an end breakpoint. This is our copy number event. And you can easily observe that the copy number events can overlap. For example, here, the first node includes a long copy number event from a star to a triangle, and then a following event overlaps with this one. This is a short event from a star to a diamond. And this is in accordance with biological truth, where, for example, we can have one chromosome doubling, and then we have shorter copy number events inside of this chromosome. What we can also observe is that the copy number event borders, so the genomic loci, can repeat inside different nodes. And this is also with accordance with biological truth, because some genomic loci are more prone to DNA breakage, and so they are more probable to become borders of copy number events. The second ingredient of our model are the corrected counts absolute difference distributions. First, we have the no breakpoint distribution and the breakpoint distribution, and we use those distributions to calculate the likelihood of our CONET. We have constructed an efficient MCMC sampling procedure that is sampling both the huge space of possible copy number event trees and also model parameters. After finishing the sampling step, we have our inferred CONET. And then we use maximum likelihood to attach the cells to nodes of CONET tree. What's important to notice here is that the cells can not only be attached to leaves, but also to internal nodes. Having this CONET and cells attached to it, we can recreate copy number change history in each genomic bin in each cell. And if we cluster those genomic bins that sell exactly the same copy number change history, we can perform copy number calling procedure. When we average the corrected counts in each of those clusters and we round them to integers. This is how we call the copy numbers for genomic bins that were changed. For all the rest of the bins where there were no copy number change history according to our model, we assume that copy number is equal to 2. This slide illustrates excellent performance of our model on simulated data. We have sampled 2 different sizes of trees. First, the tree that includes 20 copy number events and then 40 copy number events. We have then attached different number of cells to each tree. And then we have sampled the per breakpoint data using 2 scenarios. First, the well-separated scenario imitating good quality of biological data where breakpoint and no breakpoint distributions are well separated. And then the most demanding scenario with poorly separated breakpoint and no breakpoint distributions that imitate noise in biological data. The inferred tree sizes oscillate around the true value for the smaller trees. And for the bigger trees, we can see that the size of the tree is rather underestimated. We can also observe that the size of the tree strongly depends on the prior. Then, the most difficult part was inferring the exact structure of the tree. And here we have edge precision. This is a parameter that measures the percentage of the edges present in our inferred tree and also present in the true tree. And we can see that for this most demanding task, we also achieved very good results, especially for smaller trees, when we do actually infer correctly over 80% of edges. For bigger trees, we still have satisfactory results, with edge precision over 60%. And the last measure, symmetric distance, is assessing the quality of breakpoint inference. For the easiest scenario, the symmetric distance is almost 0. So this means that for almost all cells, we have inferred all their breakpoints correctly. And here, For other scenarios, the symmetric distance is equal to 1. This means that we have wrongly inferred only one breakpoint in an average cell. In this picture, you can see CONET inferred for a true biological example of 260 xenograft breast cancer cells. They were sequenced using DLP technique, which omits the pre-amplification step. This tree, as you can see, is much more complicated than the one that was presented in the synthetic example. There's a lot of information included here. First, we can see numbers printed at the beginning of each node. Those numbers signify number of copy number events that were collapsed into one node whenever it was possible. The second information are breast cancer genes printed when the copy number events actually overlapped with those genes. The third one is the color of each node. And you can see in the legend that when the node is white, this means that no cells were attached there. And the darker the shade of the green color, the more cells were attached to each node. Okay, so what information can we see in this picture? What important biological conclusions can we draw from it? First, we see this very big node that includes 59 events and a lot of breast cancer genes. This is a trunk event, so this means that all the cells underwent these copy number changes. And you can see here many known genes. For example, such tumor suppressors as RB1 or TP53, and also a very characteristic breast cancer genes BRCA1. Okay, one more thing. Underlying of each gene means that it appears only once in the CONET. Okay, so second very important information that we can see here is branching of this tree, which divides almost all cells into two distinct subclones. On the left, we have a smaller subclone that was actually driven probably by this unique anti-RK3 copy number event. And in this second subclone, we have also a setting event that includes 3 unique copy number changes in those breast cancer genes. In this slide, we can observe excellent performance of our model in its second task, that is copy number calling for the same breast cancer dataset. The upper heat map is illustrating the corrected counts for this biological dataset, and the lower heat map is illustrating the copy numbers inferred by our model. In columns, we can see genomic regions, and in rows, we can see single cells, and the cells are ordered the same way in both heat maps. The coloring is also similar for both heat maps. 0 means neutral copy number, shades of red mean amplifications, and shades of blue mean deletions. The first thing that comes to mind when we look at those two pictures is the excellent denoising of the biological signal that we can see in the second heat map. What we can also observe is that our procedure correctly infers most of copy number events, especially if they are present in enough number of cells. Also, we can see that the copy number events are inferred for very, very short genomic regions. In summary, I would like to remind you that CONET is the first Bayesian probabilistic approach for joint copy number evolution inference and copy number calling using single-cell DNA sequencing data. Our model is robust to errors in single-cell DNA sequencing data because it jointly models per breakpoint and per bin readouts. For simulated per breakpoint data, we have achieved excellent breakpoint detection and very satisfactory tree structure reconstruction, given that we used the correct priors. We have also inferred plausible events for the true breast cancer dataset, where we have tumor suppressors deleted in the trunk of the inferred CONET, And we observed further branching, revealing 2 distinct subclones in the true copy number 3. Also, we have observed smoothing of noise in biological signal. I would like to acknowledge my co-authors, Tomasz Sąkała, who implemented very efficiently our model, Wajim Yasoyedov, who had helped us with all the theoretical parts of our modeling, and first of all, Eva Szczurek, who is a really great, inspiring mentor. I would also like to thank our collaborators from Merck Healthcare in Germany and Memorial Sloan Kettering Cancer Center in USA. Thank you all for listening, and I invite questions.